I'm Dr. Sushmita. Today I'm going to discuss with you about histopathological changes in case of herpes and zoster virus. Here you can see intraepidermal blisters. Here you can see free floating keratinocytes called acantholysis, which are seen in viral conditions and immunobullous disorders. You can see that this basal layer of epidermis and other layers are separated. There will be a formation of split. So you can see fluid filled blister seen, and it is a intraepidermal blister. There are other conditions where there will be a subepidermal blisters also. And if you see, you have to basically have to differentiate between this herpes simplex virus and varicella zoster virus. In herpes zoster, usually the clinical features will follow a dermatological, means a dermatome, whereas in zoster virus, clinical features usually resides only to the lips and genitals. So thus, by clinical features, you can differentiate whether it is herpes zoster or herpes simplex virus. And other way is you can do PCR test or you can do immunohistochemistry. And other way is you can see the hair follicle involvement. Usually hair follicles are mostly involved in case of zoster compared to the herpes simplex, because usually the virus resides in the sensory ganglia. Virus reactivate along the sensory nerves, and then some nerves connect through the hair follicles. So thus this virus travels from nerves from there to hair follicles, and then it will reach the epidermis and infect the skin and keratinocytes. Thus this is the hair follicle, opening of hair follicle. So it is a condition of zoster, and thus you can differentiate sometimes histopathologically whether it is a zoster or simplex. So this herpes folliculitis is also mostly seen in zoster compared to simplex because of the same reason. In zoster, more hair follicles are involved than in a simplex. And the most important thing you have, important histopathological findings are, you have to remember 4 M's. My first M is molding. You can see all the keratinocytes are clumped together and they form a mold. So this is a molding, and there will be multinucleation, and there will be a margination of chromatin. Here all DNA is pushed outward to the wall. So there will be a chromatin margination will be seen, and there will be megacells. All the cells you can see here, all the cells are enlarged, and you can see this cell with pale slate gray appearance, which is seen in both this herpes and varicella virus. And thus you can differentiate other immunobullous disorders from viral infection.